Hi, my name is Roger Clem, and I'm an associate professor in the Department of Neuroscience. And one of the major focuses of my laboratory is on the neural circuit mechanisms of fear. In particular, we're interested in how fearful memories of traumatic events are encoded in the brain. In conditions like PTSD and panic disorder, fearful reactions to reminders of a traumatic event can be very debilitating in terms of day-to-day life. One of the reasons these conditions are so difficult to treat is that we have very little understanding of how memories of the trauma are encoded or which circuits mediate fear in response to environmental stimuli. In my laboratory, we've identified the prefrontal cortex as a critical region for encoding of memory in the mouse brain, but in addition, we've identified a specific neuronal cell type in this area that orchestrates the output of this complex circuitry to elicit fear in a mouse. It turns out that these cells are defined by their expression of a neuropeptide known as somatostatin, and we think this may be released from these cells under certain conditions. The goal of this project will be to examine whether these neurons release somatostatin in behaving animals and whether that process regulates the expression of fear behavior.
Hi, my name is Paul Slesinger. I'm the director for the Center for Neurotechnology and Behavior and a professor in the Department of Neuroscience at Mount Sinai. My lab is focusing on elucidating the molecular and cellular mechanisms underlying drug addiction. My long-term objective is to discover new therapies for treating drug and alcohol abuse. As Roger stated, one of our goals is to establish for the first time a fundamental role for somatostatin in emotional memory. Currently, there are no techniques suitable for detecting peptide release on a fast timescale in vivo. My laboratory has been working on a technology to develop biosensors that measure the release of a specific peptide in real time during behavior. To achieve this, we have created a biosensor that generates an optical signal in response to a peptide binding to it. Through discussions, Roger and I realized that a collaboration would provide the perfect opportunity to monitor somatostatin signaling in the prefrontal cortex during behavior. If successful, our new findings could open up new avenues of research for treating pathological fear disorders like PTSD. We would like to thank the donors for their generous support of this work.